Hi, we're continuing with my notes for cell biology and life science in a nutshell. We just learned a bit about the nucleus and genetic material within it. I'm just reviewing currently the um, important proteins. Well, we learned how proteins are made from transcription of DNA to mRNA, and this is done um, through through the transcription process, and um, the mRNA is transcribed into various proteins in the ribosomes, and ribosome subunits are made in the nucleolus, um, inside the nucleus. So various proteins, there's 3D diagrams here. We're just going over it. And a lot of these proteins play an important part in the cytoskeleton, which controls the shape of the cell. Um, the cytoskeleton um, consists of a um, proteins which help when the cell needs to be uh, shuttle around different molecules like genetic material, etc., as well as movement, any kind of movement across the cell involving any of the organelles. So different parts of the cell cycle. So these proteins, this one's called Vimentin, a type 3 intermediate filament protein expressed in mesenchymal cells, which we learned are stem cells that differentiate into various types of cells depending on which cell tissue it's in, playing a significant role in supporting and anchoring the position of organelles in the cytosol, so which is the cytoplasm, basically. So that's another protein. This is called plakophilin 1, a protein which is involved in molecular recruitment and stabilization during desmosome formation. And desmosomes are proteins that we learned link two different cells together. They're important in the communication and signaling, as well as connecting of cells within tissues. So desmosome is a kind of protein structure. And they also help with these um, help with these connections help with receiving important molecules um, um, from cell to cell. Plakophilin 1 is a protein which may be involved in molecular recruitment as, let's see, which one was it? Okay, this is alpha-catenin, suggested to function as a linking protein between cadherins and actin-containing filaments of the cytoskeleton. So different kinds of proteins interacting. And this is a picture of a barrel-shaped structure of the centrioles. This is for human cell. A centriole is a cylindrical-shaped cell structure found in most eukaryotic cells, and it's required for centrosome duplication in various organisms. So, and this is important when the cell is doubling or it's going through division. That's the centrioles. And the centrioles so they make up the centrosome, which are would help with DNA when you have DNA chromosomes that need to be divided. So you have two chromosomes. Each chromosome has two strands of DNA. When you're dividing the cell, each double strand separates into a single strand, and the centrosome and the centrioles, which make up the centrosome, are what help with this pulling apart of the chromosomes during the division cycle. This one is showing intracellular transport kinesin protein, motor protein. Transport molecules move across microtubules. So it's a protein that helps with movement on the cytoskeleton, or microtubules, which make up the cytoskeleton. It's again a network of fibers. The infrastructure of eukaryotic cells, prokaryotic cells, archaeans is made of this cytoskeleton. In eukaryotic cells, fibers consist of complex mesh of protein filaments and motor proteins that aid in cell movement and stabilize the cell. The cytoskeleton extends through the cell cytoplasm, directs a number of important functions such as helping the cell maintain its shape and giving support to the cell. A variety of cellular organelles are held in place by the cytoskeleton. It assists in the formation of vacuoles as well. The cytoskeleton is not a static structure. It is able to disassemble and reassemble its parts in order to enable internal and overall cell mobility. Types of intracellular movement supported by the cytoskeleton include transportation of vesicles into and out of the cell, chromosome manipulation during mitosis and meiosis, and organelle migration. The cytoskeleton makes a cell migration possible as cell motility is needed for tissue construction and repair. Cytokinesis division of the cytoplasm in the formation of daughter cells and in the immune cell responses to germs. So the cytoskeleton is important. It assists in the transportation of communication signals between cells. It forms cellular appendages like protrusions called cilia and flagella in some cells. Depending if the cell is mobile, needs to move. The cytoskeleton is composed of at least 3 different types of fibers: microtubules, microfilaments, and intermediate filaments we learned about earlier. These proteins are distinguished by their size, with microtubules being the thickest, microfilaments being the thinnest. Microtubules are hollow rods functioning primarily to help support and shape the cell, and as routes along which organelles can move. Microtubules are found in all eukaryotes. They're about 25 nanometers in diameter. Microfilaments or actin filaments are thin, solid rods, active in muscular contraction usually. Microfilaments are particularly prevalent on these muscle cells. Similar to microtubules, they are typically found in all eukaryote cells. Microfilaments are composed primarily of the contractile protein actin, measuring about 8 nanometers in diameter. They participate in organelle movement. They are abundant in many cells, provide support for microfilaments and microtubules by holding them in place. So the actin and these filaments form keratins found in epithelial cells, neurofilaments in neurons. Intermediate, excuse me. And so intermediate filaments are intermediate. They're about 10 nanometers. So halfway, about intermediate size between the microtubules and microfilaments.